Alright, today's lesson is on enzymes. We're going to take a look at what enzymes do and how they work, in particularly the lock and key hypothesis or theory. So have a look at the keywords, make a note of the keywords: enzymes, active site, substrate, and denatured. Okay, that's a quick definition of a digestive system, an organ system where organs work together to digest and absorb food. Can you list some main organs in the digestive system? Have a go. Pause the video here. Okay, how many have you got? Let's have a take a look. Okay, so the mouth, the esophagus, the stomach, the liver, the small intestines, the large intestines. So mouth is where the digestion starts. Esophagus is a tube that pushes the food down using peristalsis. Stomach, where digestion continues. Don't forget it's got hydrochloric acid. It's got the enzymes in there. Digestion continues there. Liver is an organ that produces other things as well as bile. Bile has two functions. It neutralizes the hydrochloric acid and it emulsifies the fats. And then the small intestines is where food is absorbed. It's got a very large surface area and it's surrounded by a network of blood vessels to take the digested food away. And then you've got the large intestines where water is absorbed. There are two types of digestion that is carried out by the human body. What are they? Let's see if you can write down the two types. Okay, let's have a look. So, physical digestion. This is where the food is churned by the muscles of the stomach, and this physically breaks the food up too. And then food broken down by our teeth into smaller pieces. This creates a large surface area for the enzymes to act on. Okay, that's physical digestion. And then the chemical digestion is where it breaks down large insoluble molecules into smaller ones, and this is carried out by enzymes. Make sure you know both types of digestion taking place in the digestive system. We'll then move on. So what is an enzyme? See if you can explain what an enzyme is. So, it's a biological catalyst that speeds up the chemical reaction in a living organism. Enzymes are protein molecules. They catalyze specific reactions due to their active site. Okay, they will have a unique active site that only certain substrates can fit into. They are denatured at high temperature, so the active site changes at a high temperature, which means the substrate can no longer fit into the active site. So enzymes have an optimum temperature. Also, they're denatured by a change in the pH. And again, the active site changes and the substrate can no longer fit into. So they also have an optimum pH. What I'd like to do, please, is think about how enzymes work. Look at this picture and see if you can work out how enzymes work. Use the word substrate, active site, and the enzyme. You can pause the video here now. Okay, once you've done that, let's have a look then. So you have the lock and key mechanism. This is an explanation how enzymes work. So look at the series of pictures above, and again, this time, have another go at explaining how you think an enzyme works. Use the keywords. Take your time on this. You can pause the video here. Okay. So, you've already done this, so you've described the lock and key model to explain how the enzymes work. You can clearly see there's a substrate, so there's your insoluble molecule, and it fits into this active site. That will fit there, and that will fit there. That's the enzyme substrate complex. The enzyme then does its job, function, breaks it down, and that, which was an insoluble large molecule, is now broken down to two smaller soluble molecules, and they leave. And the active site does not change, and it is ready to receive the next substrate. Have a go at this. Rearrange the sentence in the correct order to show how an enzyme works. So this will help you self-assess your explanation of how an enzyme works. So you can pause the video and have a go at this. Let's see how you did then. That was number one. Enzymes are made up of proteins and they speed up chemical reactions. Number two, some enzymes break down large molecules into smaller molecules. The substrate molecule fits into the active site of the enzyme, making an enzyme substrate complex. Only the substrate with the correct shape fits the active site. The reaction takes place in the active site, making small molecules. The product, two smaller molecules, leave the active site. This is called a lock and key mechanism, and the enzyme can be used again and again. Moving on. Here's 3 further questions for you to attempt. Have a go at this. Pause the video here. Okay, if we move on then. Match up the words with a definition. Pause the video here. This is what you should have got. Let's have a look. So a catalyst, substance that changes the rate of a reaction without being changed itself. An enzyme is a biological catalyst. Activation energy, the energy needed for a chemical reaction to take place. An active site, the special site in the enzyme where the substrate binds. And that's a review of what you've learnt today.